Hi, my name is Kacey. As Dr. Running said, I am from the University of Massachusetts Lowell, which is roughly 1,000 miles away from here. And I'm part of the REU program in plant biology. My mentor is Dr. in the biology department. This is my first ever research experience, my first time away from home, and my first time in Louisville. So I'm really excited to be here this summer doing this work. So I'll start off by telling you that Lonicera maackii is an invasive species, and to understand what it is, we have to define what an invasive species is. And according to the National Invasive Species Management Plan, it's a species that's non-native to the ecosystem under consideration, and whose introduction causes or is likely to cause economic or environmental harm or harm to human health. That means that not every species that's introduced into a new place is an invasive species. There are plenty of species that come to new ecosystems and environments that really don't do any bad damage to the ecosystem. They actually can coexist quite well. But certain species have traits about them that make them more likely to be detrimental to the ecosystem. So I'll give you a couple of quick examples that you might have heard of before. In the bottom left corner we have kudzu, which has basically taken over the south. It was introduced on purpose, but in the type of climate that the southeast United States has, it's kind of just gone crazy, and they spent a lot of money trying to remove it. And the second one, the insect pictured, is the emerald ash borer, and the larval stage of this organism is really, really awful to the ash trees because it will burrow in through the bark into the tissues that underlie the bark, the xylem and phloem, and it will disrupt the flow of nutrients in the water to these trees, and it effectively kills them. In I'm not sure if this is a yearly estimate or not, but it is 5% of the global economy is spent dealing with these species, which is $1.4 trillion, which could obviously be used for better reasons. So the invasive species that I work with is Lonicera maackii, which is honeysuckle. And there are lots of species of honeysuckle. This is the one that is the bush honeysuckle. It's native to China and Korea along the Amur River Valley region. It can be up to 16.5 feet tall, and it has a really long growing season, so it's really early to put its leaves out, and it's one of the last plants to drop the leaves. It has flowers in the early summer, and it fruits in the late fall. The fruits are spread by birds, so basically the birds eat the fruit, and then wherever they choose to excrete the fruit, that's where the new plant could potentially grow. These berries actually aren't even that good for the birds. Some have likened them to kind of bird candy bars because they're really high in sugar and they don't have a lot of good nutrition in them, so even though the birds eat them, they're not super great for the birds. The honeysuckle, when it grows up, it forms a really thick canopy, and what happens is it shades out the plants that are underneath it, and so in places where there's a lot of honeysuckle growth, you don't have a lot of growth of plants on the ground. That means that the wildlife that utilize that space is less able to find food resources because the other plants just can't compete with it. Honeysuckle was introduced to the United States on purpose in the late 1800s. It was put in a lot of gardens because it's visually appealing, and it was also thought to be good to provide cover for wildlife, but by the 1920s it became apparent that it had escaped its natural zone that they put it in, and it was starting to cause huge problems. And efforts to cut the honeysuckle back aren't always successful because unless you clear out all the parts and dig up the root ball and everything, there's a chance it can grow back. This is a map that shows the range of honeysuckle in the United States. As you can see, that's where Louisville is, where the cardinal is, and then where the arrow is, that's where I'm from, just for fun. I actually didn't think that it was invasive in Massachusetts, but surprise, we have it too. A map from 2007 did not show Massachusetts as being invaded by honeysuckle, but now it is. So that just shows you how it's able to spread. So this is the invasive range of it. And then for the studies that I'm working on this summer, this is a map of Louisville to give you some relative locations of things. You can see where the U of L Health Science and Belknap campuses are. And then in the upper left, you can see that's Twin Parks. That's where my first study is. And then kind of right-hand part of the map is Blackacre, which I'll get to after this first part. So the first study that I'm working on is to see how the small mammal community responds responds to the presence of a mar honeysuckle. This is a part of the dissertation research project of Bill Parsons, who's here today. That's Bill. And what we're doing is we're examining how honeysuckle interacts with the biodiversity, the population structure, and microhabitat preferences of the small mammals. So basically we're seeing whether the honeysuckle affects how they move through their habitats, where they like to, what kind of habitats they utilize. The site for the study is Twin Parks, which I showed you on the map. And this particular place has a variety of habitat types. There is a flood channel zone that runs through it, and so that type of area is a little different than the surrounding areas. So we can test different types of habitat to see if there's a correlation. The first thing that we had to do was measure plots, subplots, and microplots, meaning we took a grid and divided it. There's 9 subplots per plot, and then each of those is divided up into microplots. So that just helps us keep track of where we're looking. This summer, the types of things we're looking at, first we had to make the plot maps, which involved mapping the shade and the flood and the coarse woody debris, because small mammals really like to be covered up when they're moving around because they feel more safe that way. And honeysuckle provides kind of artificial cover because the predators can still see them because there's nothing on the ground underneath the honeysuckle, but they think that they're safe because to them they're covered up. So we're seeing if the small mammals kind of have a preference for the areas with coarse woody debris or not. And then what else we're doing is for each subplot we're taking a random sample of the trees and the vegetation that's found there. So we code each plant has a number, and we measure how many plants are in each of the selected squares and what kinds they are to kind of see like the species richness of the area. We also measure the honeysuckle plants that are found there because some are juveniles, some are adults, and we kind of measure the base of them, how tall they are, whether they're alive or dead, just to get an idea of the kind of habitat that's found in that particular place. For the actual part that involves the small mammals is a technique called mark recapture. And so what we do is, it's a method of trapping. There are different types of traps based on what kind of animals we're trying to attract. So we have the little kind of ones you can't see into for the mice, and then the larger ones like down here for larger animals like squirrels. And we try to trap at least 4 times a year. I've only gone once so far because the rain's kind of been a little bit of a pain. But what we do is each time we trap an animal, it gets a unique ear tag so that we can keep track of that animal. If we catch it again, then we can map where it was caught the first time versus the second time to see how it's moved through the habitat. Another thing we're doing is, we just started doing this recently, is applying fluorescent powder to them, and I'll show you a picture of that on the next slide. But what that does is, since you capture it in one place and then another place, and you can only speculate about how it got from A to B. When you put fluorescent powder on the animal, you can see exactly how it moved through the habitat, whether it went under a log or near the honeysuckle. So it kind of gives us a more accurate way to map where they're moving. Trapping the animals shows how they move over time and over different seasons. There are a lot of mice that have been captured more than once, but no squirrels, which kind of is to be expected because they have a really big habitat area. We're also in the process of building arboreal platforms to go into the actual plants to see if they're traveling through the plants that way. The one thing with this method is we need to get more captures because as it is now, the numbers are a little bit low, as you can expect. Not every trap is going to catch an animal every time, nowhere near that. So if we get more captures, we'll be able to build statistical significance, hopefully. So here are a few pictures of that, what we do for that study. In the upper left, that's the equipment we use to measure the animals and apply the ear tags. It doesn't really hurt them, it's kind of like getting your ears pierced. They can't feel it after that. And because we measure all of the dimensions of the animal, the tail length, the body length, things like that. When we catch raccoons, we don't touch those, we let those go. So that's a raccoon that we caught. It's really cute, but not really very nice. So on the bottom of the picture is us applying the fluorescent powder to the mouse. As you can see, there's the mouse. We kind of hold onto it with a plastic bag, and then we kind of just dunk it in the powder. It doesn't hurt them. And then, as you can see, it's running away, but on the ground there's pink powder, and so when you go back at night and you shine a fluorescent light on the ground, you see exactly how the animal moved, so that's kind of cool. So we're in the process of actually doing another session of that right now. And the second study that I'm helping with is actually in conjunction with researchers at the University of Indiana Southeast, a team headed by Dr. Omar Atum. And this study is looking at how honeysuckle could potentially affect the movement of eastern box turtles, which is the species Terrapin carolina. It's conducted at Blackacre Nature Preserve, which is a protected area. It's a state nature preserve. And what we are doing with that is to see where the turtles are found compared to bush honeysuckle. And there's a potential to use this information to help Blackacre make a case for why honeysuckle removal is important. They have done a few rounds of it, but it's thrown back in a lot of cases. So this study uses a different method, even though it's kind of on the same idea. It's called telemetry tracking, which a lot of you might have heard of. What we do is each turtle receives a transmitter. It's glued onto the side of their shell with epoxy, which is non-toxic and doesn't harm them. And also that location makes it so it doesn't negatively affect their mating ability. Each transmitter has a specific frequency, kind of like a radio station. And we use a transceiver, which is this device down here, to find the turtles. So it's basically like playing the game, are you getting warmer, are you getting warmer? Nope, getting colder. You have to kind of follow the signal to find where the turtle is. And so once you finally find the turtle, which can be hard because as you can see they aren't exactly neon colored and flashing lights on them, we record a bunch of different data points. We take the GPS coordinates of where the turtle was. We recorded the carapace cover, which means how covered was the shell when we found it. Sometimes they like to be completely covered by leaf litter. Other times they're just sitting in the middle of the path. That's only happened once and it was pretty cool. We record the depth of the leaf litter because turtles like to be kind of feel more secure and they like to burrow in. The canopy cover, the number of bush honeysuckle plants over and under 1 meter, basically how big they are, and the number of May-apples, because the researchers that I'm working with believe that the May-apples are not able to compete with the honeysuckle, and turtles like to eat the May-apples. The next thing we do after we take all those coordinates is we take a random sample nearby just to get another idea of what kind of habitat they're being found in. So then we use the GPS coordinates to make a map of the individual turtle's habitat, its home range, and make it easier to find the next time we go out. Right now there are about 16 adult turtles that we have in the study. This type of study is a little bit different from mark recapture. This one requires a lot more man hours because however big your data sample is depends on how much you go out and track the turtles. It's also important to keep a schedule because then you can see how they're moving in certain time intervals. So then the last thing I wanted to talk about quickly is educating the public, because I think it's really important, especially when you're doing research in a public place, that the public is able to understand what you're doing. And also the public is largely responsible for why we're able to do this research because they contribute donations and things. So there are summer camps at Blackacre right now, and they have had us go to the campers and talk to them about the box turtle, about the research we're doing, how it works, and also how we got interested in biology and the kind of opportunities we've had while we've been studying it. And it's really cool to see the kids interacting with the turtles, and they get really excited when they help us go find one. And I think it's really cool when they come up to us after and say, you know, I want to do biology when I get into college. And it's like, who knows if that's actually going to be true 10 years later, but it's pretty neat to maybe have a hand in it. So I really enjoy that. I think it's a really cool part of my work that I'm doing. So then with that, I'll just acknowledge some people at U of L, Dr. Eason and Bill, definitely, who've been really helpful to me. The Indiana University Southeast Research Team, the staff of Blackacre, who's helped us tremendously, my advisor at UMass Lowell, Dr. Carol Myers, and the director of the honors program, Jim Canning, who told me I'd be stupid if I didn't take this opportunity, and then the National Science Foundation for making the program possible, and then lastly Tech Labs, which manufactures TechDo, which made it so I didn't have to go to the hospital when I had a really awful case of poison ivy the first week, which was pretty awful. So thank you to them too. And with that, I'll take questions.